The 2018 Nobel Prize was partially awarded to Dr. Arthur Ashkin, who developed the first optical tweezers in the 1970s. He was able to accomplish this by manipulating the wavelengths of the lasers and taking advantage of the property that lasers don't have a uniform beam, with the center being more intense or exerting more force than the outer part of the beam. Because the center of the beam contains more momentum or force, when a particle enters the center of the beam, it remains there, as when it moves away, a restoring force acts upon the particle, anchoring it in place. However, the particle is still able to move inside the center, therefore two other laser beams are used to intersect at the particle so that the particle remains in one set place. To also aid with this, there is another separate bead inside the intersection of the laser beams that attaches itself to the particle being trapped. This bead is what allows the particle to be moved by the tweezers, giving a secondary reason for there being three separate beams, as the bead can only be moved in one direction with one beam present. Using this device, a multitude of microscopic objects can be trapped with the manipulation of the wavelength of the laser, such as DNA strands, viruses, and bacteria. Optical tweezers were originally developed to be a way to trap atoms, but it was not successful in doing so. The concept of trapping atoms in laser beams was derived from Johannes Kepler's theory that solar radiation and pressure can produce a force, referred to as light pressure, which was proven as true through Maxwell's equations and later expanded by Einstein, who found that the absorption and emission of photons by an atom changes the atom's linear momentum. Also derived from this concept, which optical tweezers preceded, the idea of cooling and trapping atoms with laser light was developed and achieved in 1985, which later received the 1997 Nobel Prize. Arthur Ashkin also had a small part in this project. Both of these technologies were derived from the same concepts but differed in focus of ways to trap the atom itself. However, optical tweezers were not able to accomplish this, and the method of cooling and trapping atoms with laser light was.
Light carries energy through energy instead of having mass. This energy is transferred through protons, causing light to have momentum through the relationship E is equal to pc, where E is energy, p is momentum, and c is the speed of light. The momentum of the light is very small, even when concentrated into beams. This means that they can only move very small bodies, such as individual cells or even strands of DNA. However, considering that the tweezers are designed for precision, it's better that they can only manipulate small amounts of mass. Since this momentum and the objects being moved are so small, the light does not actually require that much energy to maintain. Since the energy of the light can only move microscopic bodies, the energy output and therefore input is very small. However, even at that, the optical tweezers need to be built sturdily. While the optical tweezers concept has many physical prototypes, most revolve around using lasers guided by strong tempered lenses that need to be immune to the light, which is easy due to their size. The light just gets absorbed into a specific surface after passing through the microbead. The optical tweezers can also be used to measure distances by using trigonometry between the angles of the lasers and by knowing the focal point of the lenses. How do optical tweezers relate to quantum optics? Quantum optics are quantum mechanics applied to light. Optical tweezers can be used to investigate the quantum mechanical properties of atoms. Quantum mechanics allow for a single object to exist in two different states of reality simultaneously, as seen in the Schrödinger's cat experiment. Hence, an object at two locations can be observed collapsing into a single entity. The technical name for this phenomenon is superposition. However, this state is unstable and superpositions will eventually collapse into the one state or the other. This leads to collapse theories. For larger objects, the rate of collapse is faster. This is because, contrary to atoms or subparticles, larger objects interact more with their surroundings, which leads them to leak in heat that destroys quantum states. These are only theories since they cannot be tested, as it's difficult to prepare a large object in a superposition. However, if the superpositions are indeed destroyed at the rate previously predicted by the collapse theories, quantum mechanics as we know it will have to be revised. Until recently, this was impossible. It was discovered that particles held by optical tweezers are isolated from the surrounding environment. There was now a way to cool an optically levitated nanoparticle to 100th of a degree above absolute zero, the lowest temperature theoretically possible. The effect was similar to slowing a child on a swing by pushing at the right times. The goal is now to reach the ground state so that the object in two locations can be observed collapsing into a single entity. Moreover, modern physics relates to the wave functions and the outcome of specific waves when certain aspects of the waves themselves are manipulated. Each of the winners of the prize changed aspects of the laser waves through either amplitude, wavelength, and type of wave. In modern physics, we look at the relationships between these variables and their effect on the wave itself.
Following on, the global idea of optical traps and lasers is based on the manipulation and on the properties of light, hence directly linking it to our electromagnetic waves course. Like mentioned in the beginning, Dr. Ashkin began experimenting with lasers in the 1960s, and he based his searches on Kepler's words. Solar radiation pressure, along with solar wind, could explain how the comet's tail always points away from the sun. Maxwell's energy balance calculations further supported this theory by finding that electromagnetic radiation can indeed exert a force upon a surface. Thus, Ashkin thought that the same light pressure could be used in a lab to push a microscopic ball around. During his experiments, he noticed that the ball got driven towards the center and got trapped there. Radiation pressure of light can move objects. Light can pull. Now it's possible to control this radiation pressure to manipulate particles using a laser. So overall, optical tweezers are a single beam optical trap that exploit pure light and turn it into a microscopic force. Powerful laser light is now a microscopic tool. Previously, we saw that light carries energy through photons, conjugate to have a momentum. The majority of lasers produced now use Gaussian intensity distribution beams, which basically concentrate the light in one spot. The light intensity curve allows the microbead to stay centered in the laser and not gravitate towards the extremities of the beam. The radiation emitted from the light is not nearly close to being harmful. By design, the beam's purpose is to be able to safely extract cells and DNA for research. If the bodies stuck to the bead were damaged, the tweezers would not have a purpose in the medical field. Luckily, the radiation does not damage the organisms that are caught in the light.
Today, lasers are used everywhere in our lives. Just think of the barcode scanners at your local grocery store. In fact, they are used in DVD and CD players, cosmetic surgeries, metal cutting machines, weapons used by the military, and even in some cancer therapies. As for optical tweezers, their development has greatly contributed to fields such as biotechnology, bioengineering, and medicine. One of the greatest uses of optical tweezers in the biological industry is the laser eye surgery, where the retina gets reattached. These optical traps allow extremely precise operations on macromolecules and other biological organisms such as viruses. They continue to help us deepen our knowledge on viruses and other microbes in the light of discoveries such as the cure for cancer. Scientists are now able to sort healthy cells from infected ones and identify those that might be cancerous. Indeed, with an increase in precision, optical tweezers can reach the damaged cells without harming the healthy ones. This application is specifically used for brain cells. Perhaps in the future, this single-cell extraction will be used to further research and cure genetic disorders and other illnesses. As amazing as it sounds, the use of optical tweezers does not stop to cell sorting. A study conducted in the last years demonstrates how a single virion of HIV-1 was stabled, trapped, manipulated, and measured under high precision, a virion being an entire virus particle. Furthermore, at the single molecule level, this new technology can be applied to measure force, torque, and strain produced by molecular motors found in living organisms. With the means of this new technology, some viruses that infect archaea are now known to be powerful nanomachines. They are capable of exerting force in the piconewton range and are capable of coordinating their motors for efficiently packaging viral nucleic acid genome. As you can see, The pool of knowledge on viruses and bacteria only keeps expanding thanks to optical tweezers. But just how hard are these gadgets to produce? Due to this very specific ways that the lenses and such need to be positioned, optical tweezers can be a bit difficult to make. However, they are still quite attainable, and there are a few selective companies selling optical tweezers commercially. Although they can be extremely expensive, ranging from $100,000 to $350,000. Though it is possible that the prices may go down due to cheaper high-grade materials and lasers that are now available on the market. Nonetheless, ultimately, optical tweezers will be mainly sold for research reasons, and there isn't a high demand for them as they are really only needed in niche fields. To conclude, the incredible efficiency, precision, and capabilities of optical tweezers in different fields of science is what makes them stand out among many other scientific discoveries. The development of optical tweezer-related technology is still at its infancy and will only continue to expand from now on. Only the sky is the limit.